So this is Chris Jones from Georgia Tech, and the title is Flow Catalysis in Versatile and Scalable Hollow Fiber Reactors.
All right, we can go ahead and jump right to the second slide and start at the very basics in this interlude away from synthetic organic chemistry with a little bit of chemical reaction engineering. So I'd like to start by acknowledging my collaborators. A lot of this work, in fact, all of this work was done in collaboration with the Hugh Davis group at Emory University. And the majority of the work was done by Dr. Eric Machetta, who's a postdoc in my group. But there were also contributions from other groups at Georgia Tech. In particular, Ryan Lively and Bill Koros played a big role in synthesizing the fibers that I'm going to talk about today that are the basis for the catalytic reactors that we use. And of course, this work was funded as part of this NSF CCI, the CCHF. So next slide, please. So we're going to start really basic. And when you think about what vessel we all do our reactions in, this is probably the vessel that most commonly is used. This being a 1 liter size is a bit bigger than we would use in our typical synthetic chemistry. And if we click to the next slide, we can go to more sophisticated reactors that might allow us to do high pressure operation, have very controlled stirring on the laboratory scale. The middle one shows a dropping funnel that we might use in synthesis, as well as a reflux condenser. The one on the right allows us to go to high pressure. And so I think as a chemistry graduate student or a chemical engineering graduate student, when we think about doing our synthetic chemistry, this is the paradigm that we always think about. But when we want to think about how things are done practically on a larger scale, we have to change the way we view reactors. Next slide, please. So synthetic chemistry is practiced on many scales. We will be doing synthetic chemistry on a few milliliters scale when we're doing scoping reactions in the laboratory. But if we're going to make a commodity product, this is a high volume, low value product, like for example, cracked crude oil that is done in the catalytic cracker shown on the top left, or polyethylene, which is the definition of a commodity chemical that also would be produced in a very large scale reactor such as shown on the top left. These types of products are typically synthesized in a flow paradigm, where you're continuously flowing reagents through the reactor and producing the products that you want to use. If we now turn our attention to the specialty products, the complex organic molecules that we might be making that have a much higher value added to them, quite often these are going to be done in lower volumes. Specialty chemicals, pharmaceuticals are examples of these. And these would be done in typically batch processes or semi-batch processes, still in a much larger scale than we're used to working with our round bottom flasks from the first slide, but still substantially smaller scale and in a more stage-wise manner than the flow process shown on the top left. Next slide, please. So, however, taking that flow paradigm that totally dominates commodity chemical synthesis and applying it in synthetic organic chemistry for high value added products is unquestionably one of the growing paradigms in organic synthesis right now, both in academic labs as well as increasingly in industrial labs. So in 2010, there was a flow chemistry society that was founded, and in 2011, there was a journal founded specifically devoted to this field, the Journal of Flow Chemistry, as shown on this slide. And now there are many commercial and custom flow reactors, and I'll talk about a custom flow reactor in my work today, that are widely used in academic research and are penetrating industrial R&D as well. If you were interested in an ACS publication where you could read a wide array of papers on synthesis and flow reactors, OPRD, Organic Process Research and Development, would be a good place to take a look. Next slide, please. So let's think a little bit about the different types of flow reactors that are out there that are commonly used. In the center of this slide, there are 4 types of flow reactors that are used in synthetic organic chemistry fairly widely. And the one with the red box around it in the center of the slide is the chip reactor. And that's shown schematically at the top of the slide as well as on the bottom right of this slide. So on the top of the slide, you can have installed in a single microchip made out of, in this case, silicon nitride, in some cases made out of various glasses, where you can pump in multiple reagents, pass them through a mixing zone in order to get them efficiently mixed before going through a reaction zone where you can add heat, take heat away, add UV light, for example, if you're going to do photochemistry, and carry out the reaction in a flow paradigm on this chip. You might want to do heat exchange before or after the reaction. That's shown in the after the reaction zone. And then finally, you could take your product off. So an example of that is shown in the bottom right, where you have 3 inlets at the top of the blue part of that chip that go through a mixing zone before it flows into the circular tubular on-chip heated reaction zone, where a reaction can be done under flow conditions. And this particular reference that's referenced at the bottom is doing the synthesis of the compound shown in the center at a rate of about 7.6 grams per hour. This particular compound is a hypertension and high blood pressure drug. And it's done efficiently in this chip reactor because it helps to prevent some of the challenges associated with carrying out the reaction with the volatile amine. The on-chip reactor helps to keep the volatile amine with the other reacting components in the presence of catalysts as necessary. Next slide. Another example of a common flow reactor in synthetic organic chemistry is the tube-in-tube reactor. So that's got the one on the far right with the red box around it now. And there's 2 examples given on the bottom of this slide. On the right side, you have gas, in this case ammonia, that's flowed through the center tube, and that tube is slightly permeable to ammonia. So what that allows the ammonia to do is leak through that tube into the second tube that contains the overall rest of the reagents, in this case the dicarbonyl compound, and then the dicarbonyl compound can react with the ammonia in order to give you the final product. So you can think of this design as being essentially like a sparger. If you have to bubble gas through your reactor that you were doing in a round bottom flask, you're effectively doing that down the length of the tube by having ammonia pass through the walls and enter the larger tube where the reaction takes place. In the bottom example, you now have an enzymatic catalyst contained in the center tube, and you have gas exchange to the outer tube between oxygen and CO2, one of which is needed in the reaction and one of which is produced as a byproduct. So again, a way to handle gas-liquid contacting in these tube-in-tube reactors. So that gives you an idea of some paradigms for how flow reactors are being used today in synthetic organic chemistry. Next slide. So if you wanted to do this today in your lab, you could go out and buy commercially available flow reactors. 2 commercially available ones are shown on the top and the left. And in fact, the Corning flow reactor, I believe, almost as shown, maybe not quite that grand, is in Stefan France's group. He's shaking his head. It's definitely not that grand, but he's got a Corning flow reactor, and these can again be tubular plug flow type reactors or microchip reactors, all of which are now commercially available. Next slide. So what we're interested in doing in my group is finding ways to do synthetic chemistry and flow with novel reactor types. And we're particularly interested in seeing if we can come up with alternate paradigms for flow reactors that might offer some potential advantages. So the objectives of this particular study were to develop polymeric hollow fibers that are commercially used today on a massive scale for gas separations and apply them as new and versatile reactors for synthetic organic chemistry, essentially for the first time. And then second, to demonstrate the applicability of these fibers in several different important reaction classes with different types of catalysts, because we'd like to have a reactor where we can incorporate a wide variety of different types of catalysts. And the particular catalysts that we're going to use are represented by a solid surface. So that would be a zeolite, which is a Bronsted acid catalyst. We're going to use a very simple organocatalyst. This is going to be a Lewis basic amine tethered onto a surface support. And then an enantioselective organometallic catalyst, which is going to be the Dirhodium carbene system extensively studied in Hugh Davis's group. We're going to do types of reactions, a very simple deprotection. We're going to do a very simple coupling reaction, and then a more interesting enantioselective coupling reaction using the Dirhodium carbene system. So next slide, please. So how are these fibers made, and how are we able to make them on a large scale such that we could use them very simply? So on the right side, on the top, you'll see what's supposed to be depicted as a fiber cross-section. So you've got the gray fiber where it's been sliced in half, and so you can look at that cross-section, and the green part represents the fiber walls, and the yellow part represents an open center that you could flow fluid through. So you can think of it as a drinking straw. And so we're going to have a typical flow reactor. You would just flow through the center of the fiber as shown here. Now these are used extensively in industry to process separations of gases or liquids. So you could flow a mixture through the center of the bore of the fiber, and maybe some components of the mixture can preferentially diffuse through the walls and out the walls. Others that can't diffuse through the walls are going to flow through the fiber and come out the other end of the bore. And these are made by taking a polymer solution, and these are simple polymers, polyamides, polyamide imides. What we're going to talk about today is cellulose acetate, which is a very common, commercially available, low-cost polymer. And this polymer is solvated in an organic solvent, which is then passed through a spinneret. That fiber that's extruded through that spinneret crashes into a water quench bath, which helps to solidify the fibers. And then the fibers are taken up in a rotating drum. And this approach allows you to make, meters per minute of fiber very cost effectively. And as an example of how they're used in separations, on the bottom right, you see a somewhat blurry image of a man standing next to a whole bank of fiber modules. And this is in a Japanese water purification plant. And these modules are collections of thousands of fibers contained into this cylindrical module, through which you will pass, in this case, for example, dirty water through the bores, potentially, and then have some components that diffuse out through the walls, other components that come out through the bores, and that's how a separation would be effective. So next slide. So we'd like to demonstrate this type of flow reactor, and not necessarily for any particular separation. And so we're going to have, again, the cross-section of the fiber, which is shown schematically in the center with the green sphere with the white hole in the center. The white hole is your flow reaction zone, the bore, and the green donut shape is the fiber wall, cellulose acetate. And contained within that fiber wall, we're going to embed a whole bunch of silica nanoparticles or other oxide nanoparticles. So it's a composite fiber where the walls are made up of cellulose acetate polymer as well as porous silica or other porous oxide materials. The bottom left shows you a scanning electron microscope image of what this might look like. You can see that there are very open spaces of highly porous space within the polymer, with pores on the scale of 0.5 to 1 micron in size. This allows very rapid transport of liquid reagents into the polymer, or in our case, through the polymer. And then also in that image, you'll see tiny little white particles. And those white particles, in this case, are zeolite particles, which is one of the catalysts that we use in this study. So on the bottom right, you'll see that the picture of a whole slew of fibers held between one person's hands. They're flexible. They're flexible enough that you can tie them in a knot like you would tying your shoes. And on the top right, you can see for scale approximately how big these fibers are. So this is next to a typical laboratory syringe. So the bore of the fiber, the thickness of the fiber is not that different from a typical laboratory syringe that we would have. So the way we're going to use this in our reactions is we're going to use it as an ultra-short catalyst bed by flowing through the walls of the fiber and then out the side. So in the schematic shown in the center, we have some reactants, R1 and R2, labeled by spheres that are going to be fed through a sample loop and then into the bore of the fiber. And then the other end of the bore of the fiber is capped, so you can't flow out the other end. And what that does is it forces the reagents through the walls of the fiber out into a sweep flow of solvent that carries the product away. So essentially, this is an ultra-short bed of catalysts that the reagents flow past, giving you a very, very short contact time and allowing you to effectively control things like temperature associated with the very exothermic reaction that this carbene chemistry is. So next slide. So we started with two simple reactions, very, very simple reactions, just to show the versatility of the fibers. First, we incorporated zeolite ZSM5 into the fibers. So during the process of spinning those fibers from the solution going through the spinneret into the quench bath, we included in that polymer solution zeolite particles. And those are then embedded within the walls. And zeolites are Bronsted acids. So they're strong Bronsted acids, and they can do simple chemistry that a typical Bronsted acid would do. So in this case, we're doing a very, very simple reaction, deprotection of, in this case, benzaldehyde dimethylacetal to give us our unprotected benzaldehyde. And we can do this very effectively by flowing the reagents through the fiber at relatively mild conditions, 60 degrees C in this case, and get essentially quantitative conversion of our product. So we can use the surface of the particle as the active site. That's what this is demonstrating. The next example is a simple organocatalytic reaction. So now we've taken that nanoparticle, which is just now silica instead of zeolite ZSM5, and we tethered to it a simple alkyl amine group, an aminopropyl group. So you can think of this in the context of a lysine side chain and a protein. This is a common basic active site for nucleophilic chemistry. And this is a good base catalyst for the Canova-Nagel condensation, which is the simplest, easiest to catalyze, base catalyzed reaction that I know, to create a new carbon-carbon bond with an activated methylene compound, in this case melanonitrile, and the same reagent we used at the top, in this case benzaldehyde. So again, we can do this under mild conditions and get essentially quantitative conversion by passing through the walls of the fiber reactor and quantitatively recover our product. Next slide. But what's more interesting is if we can do a more advanced and a more complex reaction. And so in that context, this is where our collaboration with the Davis group at Emory was really useful. So over the course of the last several decades, the Davis group has worked extensively on these dirhodium carbene complexes, which are outstanding CH activation catalysts. And the particular system that we worked with has been well studied for a long time. It's the rhodium DOSP system with the DOSP ligand shown to you here on this slide. And this D2 symmetric catalyst is set up such that there's an alpha-beta alternating arrangement of the side chains on these DOSP ligands that is shown by the blue part of the diagram on the top right, with the planar disk essentially representing the dirhodium where the carbene will exist. And if you use this type of catalyst with a donor-acceptor carbene, and in the diagram, the donor-acceptor carbene has an aromatic group on one side and an ester group on the other, this can ideally position the carbene such that it can only be attacked from one side by the substrate. So you have the access to the carbene blocked by two of those blue partitions essentially standing up. And then you have the ester group blocking access from the other side. So one more click on the slide, and you'll see that the electron withdrawing group offers you that steric control and blocks one access. And you really only have the green arrow that provides you access to the active site in this system, giving very, very high levels of selectivity and CH activation as well as related chemistries. Next slide. So we chose to use this system as our baseline system for doing an enantioselective catalyst and for a CH activation catalyst. And so we were particularly interested in this because it's a relatively expensive ligand and it's obviously a very expensive metal. So some of the limitations with dirhodium catalysis include that these catalysts can be challenging to recycle. They can be expensive. The reactions with the carbenes are highly exothermic. You liberate nitrogen gas and you have to deal with that in your reaction. And these metal carbenoids can deactivate under some conditions quite easily. So by using an appropriate reactor, in principle, we could try to make recycling easier. We could potentially facilitate flow for large-scale production. We could have improved heat and mass transfer, since these are very exothermic reactions. Potentially getting rid of the heat as well as getting rid of the nitrogen produced are useful. And in this first manifestation, we chose to use this very short catalyst bed with flow through the walls of the reactor, as shown on the bottom right, as a way to give us a very short catalyst bed, to give us a short contact time of the reagents and the catalyst, and hopefully limit the amount of exotherm that we would see. So schematically, you can see we flow through the bore, and then we have flow out through the walls of the fiber in order to give you the good reagent catalyst contacting, where our rhodium DOSP catalyst is going to be tethered to those silicon nanoparticles. Moving forward, what we'd like to do is to work with some of the more advanced catalysts that the Davis group has developed, such as the one shown on the right that incorporates the cyclopropyl group into the ligand. Next slide. So we first had to make an immobilized catalyst, and Katie Chapiga from the Davis group developed a simple pathway to do this. We could go ahead and begin to assemble the ligand in the standard way that would be done in making the DOSP type ligands, except now we're going to substitute a functionalizable group at the end of the chain. And in this case, we decided to start with an olefin because the olefin gives us many different routes that we can use to put on a functional group that we could then use to attach this catalyst to a surface. You can imagine doing thiolene click chemistry with an appropriate olefin. You can imagine doing olefin metathesis. In this case, we did the simplest possible thing, which is just to use radicals to couple the double bond on the end of the rhodium-dirhodium complex with a styral group that's been tethered to the silica surface. So we'll take the silica particles in that fiber, first functionalize them with reactive styral groups, then use a radical coupling to introduce this dirhodium catalyst. The other thing I'd like to point out about this dirhodium catalyst is only one of the 4 ligands has been exchanged with this functionalizable olefin N group, and the other 3 ligands are the standard DOSP ligands. And so this gives us a system where the ligand sphere around the metal center is very close to the rhodium tetradosp system that's the baseline system used for so many years in the Davis group. Next slide. So the first reaction that we decided to use was an enantioselective cyclopropionation. We picked one substrate to use, shown in the blue or purple, and then we varied several different donor acceptor diazo compounds in order to generate different carbenes. Next slide. So I show you here an array of products that we prepared, and you'll see that the yields for all of these products, ranging from about 60% up to about 80%, are fairly comparable to the yields that you would obtain in batch. So the yields on the top, 77, 77, 61 for the first 3, are compared to the batch reactor homogeneous catalytic results that you would get if you just used the DOSP catalyst under homogeneous conditions. And so they're typically modestly lower in flow, although a few examples are comparable or better. And if you look at the enantiomeric ratio as well, you'll see that these catalysts are typically quite selective with enantiomeric ratios on the order of 97 to 3, 96, 98 to 2, etc. And we're fairly close to those values in the flow system as well. So putting this catalyst onto the particle and passing the reagents by the catalyst with a very short contact time has not diminished the utility, the synthetic utility of this approach. Next slide. 2 clicks. So we were also interested in doing a CH functionalization reaction, and this is a very well-studied reaction in the Davis group. It's a combined CH functionalization followed by a cope rearrangement. It again uses a typical donor acceptor carbene associated with in this case, the tethered dirhodium catalyst to produce the product shown on the right. And in this case, again, we're getting yields above 80%, enantiomeric ratios of 94 to 6, which are not far off of what you get under homogeneous conditions. So we're able to take a relatively sophisticated catalyst in this case, and put it into our fibers in order to enable reaction under flow. Next slide. So this is only worthwhile if this makes the catalyst stable and can be used for a long time. And so I show you here a plot of the percent of the major enantiomer as a function of the turnover number for the cyclopropionation reaction shown at top, our standard reaction. And you see for 2 different fibers, so we made 2 batches of fibers to make sure this was reproducible and that the fibers behaved in a uniform way. And then you see the data points that represent different amounts of reagent flowed through the system over a consecutive time period. And we probably could have gone much longer than this, and the 1,000 turnovers probably best measures the patience of Eric and Solomar in doing the reactions. And so it's quite a stable system, and we were able to get a reasonable number of turnovers on this system. Next slide. Is the catalyst robust, and can you continue to use it ad nauseum over and over and over again? A fairly simple, although not rigorously conclusive test, is to look at the diffuse reflective UV vis of the catalyst before and after use. And the dirhodium signature stands out quite clearly at 600 nanometers. And you can see after using this system for a long time, that dirhodium signature is still there. Although we should point out this is not a quantitative measure, it's qualitative. And it says that some of our catalyst is intact, but it doesn't necessarily indicate that all of our catalyst is still intact and turning over. In order to probe that, we probably have to do some poisoning experiments. So next slide. So what are the next steps? Where are we going to go next? So we're going to use a more traditional fiber flow-through reactor where we don't flow through the walls. And so what we're going to do is we're going to take the same type of design and coat it on the outside with a red layer that doesn't allow any mass to be transported through the walls. And so this was originally a figure from a separation system where we had sorbents in the walls. Now we're going to have catalysts in the walls. But these walls are highly porous. I showed you pores on the order of microns in size. So as we flow reagents down the center of the tube, some of these reagents are going to flow into the walls, contact the catalysts, and be converted. And then we'll collect all the reagents coming out of the back end of the fiber in this case. And so this is the more traditional approach to a flow-through, plug-flow type reactor in a fiber system, except we're going to now do this in this polymeric oxide composite system for the first time. Furthermore, we're going to adapt a better catalyst that's more active, more selective. The catalyst shown at the bottom, which is one of the several state-of-the-art catalysts that now exist in the Davis group at Emory. Next slide. And finally, I want to leave you with the idea that you can do very complicated multi-step organic synthesis in flow. And this work is not from our lab, but rather it's a recent paper in Nature that shows you that you can do multi-step synthesis of relatively complex pharmaceutical products in flow. So these are multi-step syntheses of chiral compounds that are anti-inflammatory or anti-tumor molecules shown on the bottom. And they were doing this on about 1 gram per day in flow, getting the product in about 96% EE. The system was able to be run stably and continuously for a week. And they used all commercially available materials in this synthesis and didn't have to do any inline separations in the process. You can do separations in between reaction zones if you hook together multiple reactors in a flow system. So I think this is an emerging paradigm that you're going to see more and more of over the years in synthetic organic chemistry. And I still think that the field is at its infancy, and there's a lot of novel work that's yet to be done. Next slide. So to finish up, I'll just acknowledge my group and acknowledge again NSF for support of this work. It's been really interesting for me as a chemical engineer to be able to collaborate with synthetic organic chemists of the highest order. I think my group has learned a lot from it. And as a quiz for everybody, can you look at my group and immediately tell that they're chemists and chemical engineers? Any idea how you could tell right away? They're assembled in the color spectrum, so they self-assembled in the color spectrum, so they're all nerds at heart. And with that, if there are any questions, Hugh, I'd be happy to take them.
So thanks very much, Chris. I need to make a comment first. You mentioned that work in these rhodium catalysts had gone over several decades. I just want to know what you mean by several decades.
I didn't mean to insult your youth, but I'm pretty sure you were working with these while I was still in college, if not high school.
Okay, now we got a more serious question. So a question from Keist. South Korea. Have you ever compared the residual rhodium contents in the final products from batch and fiber flow?
Right, so we have, and we don't detect really any rhodium in our products after the fiber flow system. Elemental analysis of the fibers before and after, as well as the liquid media that contain our products before and after, really don't show any loss of rhodium, so no loss from the fibers and no significant rhodium in the liquid solution. When we compared it to the batch case, of course, you can always do a workup where you use the appropriate chromatographic methods to remove the rhodium. You would have to do that in the case of rhodium because you can't afford to lose any. But certainly, this system appears to be quite robust towards rhodium contamination.
Okay, a question from me. So, when one is doing some of the ion catalyzed CH oxidation, and we heard great talk from John Hartwig about the CH acidation. One of the challenges is to get the reaction to go to completion without getting overreaction. Do you think the flow technology can help in terms of dealing with those types of reactions?
It certainly it depends on the nature of the kinetics and the nature of the reaction pathway. Certainly you have some additional tools in your toolbox if you want to employ flow reactors in order to deal with these types of issues. One of the other advantages of using these types of catalysts where you're isolating the catalyst onto a surface It allows you to mitigate against some bimolecular deactivation pathways where you have two catalysts that come together and deactivate by forming a dimer. So there's a lot of potential advantages, but frankly, there are a lot of potential disadvantages as well when you think about the added complexity associated with working on one of these systems. So I certainly don't want to say that the flow approach is a panacea and it's going to solve all problems. It will solve some problems, but frankly, it will create some other problems as well.
Okay, one other question from Ohio State. Are there phase separation issues using these silicate polymer blends at elevated temperatures? And what is the TG of these polymers?
So these are glassy polymers. So we typically work exclusively with glassy polymers. So the polyamide imides and the polyamides and cellulose acetates have TGs all above 100 C. That's part of the skill set and the art that is contained in our collaborators lab. This is Lively and Koros at Georgia Tech that have the ability to spin these composites with frankly remarkable amounts of silica materials in them. So some of the fibers that we have used have 50% by weight silica contained within the polymer. So half polymer, half silica, yet they retain the flexibility of the polymer such that you can still tie them in a knot and they don't break. And so this allows us to get very high catalyst loadings if we choose to by going, up on the order of 50 to 60% by weight silica, and then decorating the surface of those silicas with either our organometallic or organic catalysts, or using the surfaces themselves as active sites if we want to go the Bronsted acid route.
And one final question. This is from the Trost group. What is a typical solvent for the sweet flow? Maybe you mentioned it, but we didn't catch it.
Typically, we're using hexanes, and our sweet flows are typically used in this first study at relatively high flow rates just to make the collection of all of our product relatively easy. If you were going to actually use this for production, you would spend a lot of time optimizing those flow rates such that you don't over dilute your system too much that you waste a lot of solvent. In this first demonstration of concept, we just wanted to show that the fiber system worked. And frankly, if we were going to use this for as a means of producing compounds, we wouldn't do the flow through wall approach. We would use a more traditional approach of flowing through the center bore, collecting the products at the outer end.
Okay, Chris, thanks very much. A wonderful talk. Adds a nice bit of breadth to the whole program.